The HC11 and EpH4 cells are mouse breast epithelial cell lines which can differentiate in culture. At the same time, these cells can be transformed by a number of oncogenes. These cells are ideal for the study of the interrelationship between differentiation and neoplastic transformation. These techniques can be used in signal transduction studies to discover targets for cancer therapy. For example, the small GTPase RAC and other molecules may trigger transformation of breast epithelial cells when expressed to high levels, but surprisingly, at low levels, they may induce differentiation. Other signal transducers may behave in a similar manner. The principles may apply to other types of differentiation as well, where cell confluence plays an important role. For example, differentiation of adipocytes or myotubes. Someone performing this technique for the first time should keep in mind that plating of the HC11 cells is important. This is because cell-to-cell contact is important for differentiation. Another point to watch out is that proper extraction of the EpH4 cells from the matrix is critical for protein quantitation because any residues will affect protein determination. Visual demonstration of this method is useful because some details of the protocol are difficult to describe on paper.
Start by preparing a bottle of 50 milliliters of HC11 cell medium according to manuscript directions. To plate the cells, aspirate the medium from 5-6 centimeter 50% confluent petri dishes and wash the cells with approximately 1.8 milliliters of trypsin using a sterile 9-inch cotton-plugged pasture pipette. Then add 200 microliters of trypsin per plate and swirl the plate to dislodge the attached cells. Observe the cells under phase contrast microscopy with a 4x objective.